respectable chairperson, professor, and all the distinguished guests. I will be presenting on the most common preventable, partially preventable surgical disease in the pediatric population, that is meningomyelitis. I will be dealing with the embryology and genetics part. Misconceptions are there regarding the identification, classifications, and then the surgical repair. So early neural development embryology is required for the better understanding of the meningocele. So I will be dealing with these topics. So we all know that after a number of cell division, there will be the blastocyst formation, which consists of the number of the cells, and then there will be formation of the epiblast and hypoblast. There is formation of the epiblast and hypoblast, and then there is the formation of the precaudal plate at around day 30, and this precaudal plate is the first morphological structure that suggests that it is the cranial end and the caudal end is the next end. So now then there is the development of the primitive streak, and in the primitive streak, after the formation of the primitive streak, this epiblast will form the three layers by displacing this hypodermis layer, and it forms the epidermis, mesoderm, and then the endoderm. And if there is a failure of this layer, then there might be the pathology like neuroenteric mesenchymal cells, which forms the notochord, and just below the notochord it will form the neural tube, and then there will be in between there will be the formation of the endomesenchymal tract, which ultimately will form the structure like this, that is the diastematomyelia. So this is the importance of one of the importance of embryology. So we should know about the Hansen's node, which is a very important structure. I will be repeating about this Hansen's node very much. And here also we can see that the prospective notochordal cells are being producing the cells under this Hansen's node. So now this Hansen's node is the area where there will be the localization of the prospective neuroectoderm, and the cells are arranged in a linear cranial caudal sequence, so that the forebrain becomes the cranial part and then the midbrain. And also there are the theories which suggested that the cells are formed in the multiple neuroaxial layer. So here we can see the shaded color which can be found in the cranial as well as the caudal end. So the formation of the notochord, at first there will be the notochordal process where there will be the formation of the neuroenteric canal, and then after that the notochordal endodermal fusion will be there, and after that there will be the exhalation where the notochord will separate from the underlying epidermis layer. So that is the exhalation. So next. Now the main part is the neural tube formation, where just above the notochord there will be formation of the neural plate, which later neural group will be formed, and then they will separate from each other to form the neural plate, and then mesenchymal cells will lie in between. So if there is a failure of the fusion, and then there will form the myelomeningocele, and this neural plate will form the neural placket, which is the failure of the fusion, and this is seen in case of the open myelomeningocele. So this is the neural placket which we will be seeing at the first outer layer while performing the surgery. So these are the three fundamental concepts of neurulation, which is very important. And there was an experiment done where the cheek embryos are treated with the cytosalacin D, and this disrupts the myofilament. So when it was treated with this cytosalacin, then it was found that the embryo was consequently dystrophic. So similarly, in the neural fold fusion, there are the interaction of these cell proteins, and they have found that if there is the SPS plus mouse mutant, then there is the absence of this glycosaminoglycan expression in these cells. So here the cells are being arranged from the central to the cranial, and then it goes caudally. So in the right picture we can see the myelomeningocele, which is a failure of the caudal neuropore. Similarly, there are other theories where it suggests that there is the multi-site closure theory, where there will be formation of the 5 waves, and among these the 4 waves will cause the failure cranial neural tube, and if there is a failure of fusion of this wave, then subsequently this type of the defects will be formed, like encephalocele and cervical lumbar meningomyelocle. These all depends on where depends on the site of the failure of the closure of the neural tube defect. So next. Now the closure in detail, there will be at first the apposition and then the fusion of the cutaneous ectoderm will be done, and after that only neuroectoderm fuses, so that later this ectoderm and the neuroectoderm separate, which is called as the disjunction, and after that this mesenchymal cells will lie in between. So next. So if there is a failure of the fusion, if there is already disjunction, what happens is surface ectoderm will be fused, but in case of the fusion of the neuroectoderm, mesenchymal cells will transfer to that area, so that there will be formation of the closed spinal tube defect. So we will be having the defect like intradural lipoma, dormal sinus fistula, ventriculitis terminalis. So in particular, the cell adhesion molecules are very important for the differentiation of the cutaneous ectoderm and the neuroectoderm, where during the process of the neurulation, epithelial cell adhesion molecules will be expressed by the neuroepithelial cells, so that there will be formation of the neuroectoderm in the one side, and then the prospective epidermal cells will form the epithelial cells. So after primary neurulation, secondary neurulation is important, where there will be the formation of the caudal congenital cells, which gives the pluripotent cells, and then there will be the cavitation, which fuses with the primary neural tube. And after that, there will be the formation of these structures like phylum terminale, terminal vertical. So if there is any pathology including phylum terminale, caudal agenesis, sacrococcygeal pterygium, this is all because of the failure of the secondary neurulation. So now spinal neuroshield, there will be the occlusion, and cranial to this occlusion, it may play, there will be the initial expansion of the brain and the ventricular system. And if there is the closure of the neural pore, then here we can see that it has isolated the cranial ventricular system from the environment to a closed field space, and this there will be the pressure differences. So due to this intraluminal pressure, it provides a driving force for the brain enlargement. So the experiment was done. If the intra and extraluminal pressure are equalized, what will happen? It has suggested that the brain expansion will be significantly impaired, and then the neural tube will be collapsed, so that this is important because this is proposed as a mechanism for the genesis of the canal malformation in open neural tube defect. So next, we all know that the rate of the growth of the vertebral column will exceed the spinal cord, so the conus medullaris must have to ascend. So if there is a failure of the ascent and the conus lies below the L2 level, then it is called as the phylum, it is known as the tethered cord. So patient presented with the findings like bowel and bladder involvement, and then the paresthesia. So next is the molecular biology. So the embryonic development is also developed at the genetic level. Either the proteins will bind directly or indirectly. So ASOC protein is a signal transduction protein, which is very important. At first, this ASOC protein will bind to the past, and then it will form the Wingel's protein. It will activate the Wingel's protein. This Wingel's protein further activates the catenin. Then there will be the formation of the positive feedback loop. So due to this positive feedback loop, there will be the rostrocaudal metabolic patterning in both the vertebral venous system and the spinal column. So this pathway is also important for the control of the differences of the ventral horn motor neurons and the molecular substrate, also for the median hinge point. So here we can see that in case of the median hinge point, this causes the formation of the actin fibers. So there will be the neural folding defect if there is the defect in the sonic ASOC gene, and if there is a point mutation in the sonic ASOC, if there is mutation in the sonic ASOC gene, then what will happen? Then the patient may also present with the defect like holoprosencephaly. So now the transcription factors which helps in the transduction of the structures. So it also modulates the transcription of the other genes. So Hansen node expresses the various transcription factors like glucosamine, omidomin, protein, OTX2. Similarly, Hansen node also expresses the proteins. So next. So if in isolated ectoderm, it will form the neuroectoderm rather than the epidermis, but there is a pathway, that is the BMP4 proteins expressed by the ventral cells, and this will inhibit the formation of the neuroectoderm, rather it forms the cutaneous ectodermal cells. So this is the work of this Hansen's node, which produces the factors that will inhibit the BMP4 factors, and then this will cause the formation of the neuroectoderm rather than the epidermis. So another functions of the Hansen's node is the formation of the molecular species, and there will be the formation of the factors that specify a more rostral phenotype and the more caudal phenotype, and there will be the interaction of these two signals. Other involved genes are also there. Other involved genes like PAX3, Omebox genes, these all are important for the formation of head formation, notochord. These all genes are involved. And so next, if there is the expression of the earlier genes, then there will be the formation of the prosencephalon, mesencephalon, and the myelencephalon. And the later, the various Omebox genes will be expression due to these genes which controls the metametric segmentation within each segment. Now the main embryogenesis, as there are mainly two theories, that is the non-closure theory, where there will be the formation, where there will be the failure of the formation of the neural tube, and in over-distension theory, it has been suggested that the neural tube has closed, but again, and there are formation of the multipotentials which are responsible for the various normal and abnormal soft tissue types like ovarian and renal tissues, Wilms tumor or teratoma. So we have to look for the other causes of the, we have to look for the anomalies while performing the surgery for meningomyelocle. There are also genetic components involved, where we can see that there is different concordance rates, Mendelian inheritance, chromosomal rearrangement, all has been seen in the neural tube defects. Similarly, there are misexpression, there is first mutation, there is a deficiency in WNT5A gene, and also there are the defects in various genes which is responsible for craniorachiasis, and then neural tube defect, spina bifida. These all are due to the various genes involved. Next. So next. But now the particular interest is due to the one of the lady has found that in 1722 and 1732 the role of nutrition in the embryogenesis of neural tube defect, because the crops, there was no folate during that area, so the neural tube defect has rise. So she found out that due to the folate deficiency, there is the more chances of the neural tube defect. So since that time, there has been working about the genes that control the folate metabolism. There have been thoughts ongoing about the role of the pathways due to the folate. So mainly there are two genes that will be mutation. One is the 5'10" methyl tetrahydrofolate reductase, and the next gene will be the methionine synthase. So what we'll do is there will be decreased amount of methionine and increased amount of the homocysteine. So due to this disorder, methionine metabolism will be there, which cause the neural tube defect. So if we ingest higher dose of the folate, then it will overcome the relative deficiency by restoring the more normal homocysteine level and methionine level. So next is the valproate, which is a drug that also produces the neural tube defect, and due to this, there will be the it usually interferes with the tetrahydrofolate conversion to the 5'10" methylene tetrahydrofolate, and it also causes the folate dependent methyl, and other affected genes are also there, like which includes the non-genetic factors, nutritional factors, and due to hyperthermia also there is aberrant expression of these genes. So genetic involvement is very important in case of the neural tube defect. So neural tube defects is heterogeneous in origin. It represents the end result of the various embryonic disorders, and genetic models require more advancement and more research are there. However, early neural development predicts the anatomy of the meningocele, and knowledge it certainly helps us to repair for early repair. However, exact mechanism remains a mystery.